Hello, my name is Alicia Henn. I am Chief Scientific Officer of Biospherics, and today we're talking about eliminating edge effect in 96-well plates through control of the temperature of the cell plating environment. So here's your standard 96-well plate. This one has A549 cells that have been plated in it, and I'll do it here, and then they're stained with crystal violet just so we could look at the distribution. And they were plated under traditional cell culture thermal conditions where they're plated at room temperature and then moved to a 37-degree incubator. And you can see the sort of weird pattern of non-random distribution of the cells in the wells that are closest to the edge of the plate. And you can actually see a little bit of this on the next row in as well, if you look. And this is your classic edge effect. And there's been lots of different explanations for why this happens. Some people blame vibration in the incubator. Some people blame evaporation around the edge of the plate. And there's also this model of thermally produced edge effect, in which you get convection currents in these edge wells due to temperature variations between the center and the edge of the plate that drives these cells towards the edges of the plate. So we're talking about this thermal model today and seeing whether we can eliminate it by eliminating those temperature changes. Now, the way most people deal with this edge effect is just to eliminate those edge wells from the assay. Fill them with buffer, don't use them, only use the 60 wells on the inside of your 96-well plate, then for the assay, eliminating the 36 wells on the outside. Of course, this is terribly wasteful. You're losing 37% of your assay space, $119 million wasted annually just in the plastic for the 96-well plates, not to mention all the other reagents and all the time that goes into plating all this. So this is actually a pretty impactful issue. Now, there was a paper published about 20 years ago that showed that if you kept the temperature of these plates at room temperature for 1 to 2 hours before you put them in the incubator, you could reduce edge effect for cell-based assays. But of course, this is terrible for cells. Cells do not like being at room temperature. They like being at physiologically relevant temperature, 37 degrees. And so we decided to take on the question of, can we prevent edge effect by plating cells at constant 37 degrees C? Now, Biospherics, we make Xvivo systems which control the cell handling environment. We have incubators. These are these black doors in the back that open straight into a controlled cell handling space where you can have constant CO2, constant oxygen, constant HEPA filtration of the atmosphere, and you can have 37-degree floor as well as 37-degree atmosphere, so the cells are never exposed to highly variable room air conditions. So with these controlled conditions, we are able to set one processing chamber at room temperature and set another one at 37 degrees and see what happens when we plate cells in each condition. And we also used the HoloMonitor M4 holographic microscope from PHI to look at the effects on cells plated at these different temperatures. So here I'm going to switch to a video and show you we are able to use a thermal imaging camera to look at what is happening to the actual temperatures in these different conditions for plating cells. So on the right-hand side, the pink side, everything is at 37 degrees, so it's all the same color. The pipetter, the tips, the reservoir, the medium, the plate, everything's at 37 degrees. Whereas over here on the left, we're plating in room temperature conditions, like a biological safety cabinet. And you can see that the plate is cool, the background is cool, the medium is warm. That was warm to 37 degrees before we started, but it's cooling quickly around the edges of the reservoir, it's cooling in the tips, it's cooling very quickly in the plate. And now as this plate gets finished, we're going to speed up this video a little to show you how long it takes for that plate to cool all the way off to room temperature. Because sometimes you're not plating the same suspension of cells all the way across. It may take you a few minutes to fill that plate. And even if you're using an automated robotic system to fill your plates, if you're doing it at room temperature, the same thing is happening to your cells. So we're at 5 minutes now, and that plate is almost at room temperature, 6 minutes. And it's really at your room temperature. So what happens when these plates go into the incubator? Now on the bottom we have a plate that was plated at 37 and incubated at 37. So that hasn't changed temperature at all. But here comes our plate that was plated at room temperature. And you can see, of course, that it is much cooler than the surrounding environment. And I'm actually going to speed this up because it takes 12-13 minutes for that plate to warm all the way up. But you can see that the edge wells, of course, are warming up first, and the center wells are staying chilled longer. So by the time we get to here for 5 minutes, those edge wells are almost up to temperature, but the center wells are still cool. And this differential in temperature could be the sort of thing that would drive those thermal currents that would push those cells around differently in the edge wells than in the middle if they're still settling down during this time. And it does take cells a good 15 minutes to settle down to the bottom of the wells. So this is a highly relevant time to have these thermal currents actually influence the distribution of cells. And I'm going to speed this up so you can see it's at 13 minutes now, and that plate's finally warmed up to 37 degrees. So now let's look at what's happening to the cells. And this is time-lapse imaging done with the Holomonitor M4. And let me set this up for you before I let it play. We're looking at the cells in the wells of E12 and F12 on the plate. So these are the right-hand edge of the plate. And on the left-hand side, we have ones that were plated at room temperature before they were put in the incubator on the Holomonitor. And on the right-hand side, these are pseudo-colored pink rather than blue. These ones were plated at constant 37 degrees. Of course, the same temperature as they're being incubated at. And these are cells that are settling down into the bottom of the well and coming into the focal plate of the microscope. So it's like you're lying on your back making snow angels in upstate New York, the way we always do, and the snow is just falling straight out of the sky and settling on your glasses. This is what the angle we're looking at these cells at. So I'm going to play this, and we're going to see what happens to the cells as they settle down and hit that bottom of the well. And you can see over on the right, these cells settle straight down and start attaching right away. On the left-hand side, they actually are rolling across the floor of the well as they settle down. I'm going to play that back again. You can see that motion as it goes back, and then it goes forwards again. The cells are rolling down into the right, which makes sense for the position of the wells in which they're in. I'll play that one more time so you can see that rolling motion. And the only difference between these two sets of wells is the temperature at which the cells were plated before these images were taken. So let's go back to the slides, and let me show you some cell tracking analysis that can be done with the Holomonitor software, in which we look at cell motion. And we can trace the motion of each one of these cells over the time lapse. And we can see with a combined vector of all these vectors, you can see a very strong combined motion across the well as those cells settle down. And that vector actually points to right where the well is on the plate, which is consistent with a thermal gradient between the center of the plate and the edge of the plate. Whereas the same well, a plate that was plated at 37 degrees, you get random motion of the cells that all cancel each other out, and you end up with a combined vector that's near the origin. So when you plate the cells at room temperature and put them in a 37-degree incubator, just like you do in a traditional cell culture lab that's open to the room, you do get this edge effect. When you plate the cells at constant 37 degrees C and then put them in a 37-degree incubator, you can eliminate this edge effect. Those cells are randomly distributed all the way out to the wells around the edges of the plate. So it doesn't have to do with vibration or other things. This is really a temperature-driven effect. So because we could with the Xvivos, we decided to reverse that temperature chain and see if we could get a reverse edge effect, and we did. If you plate cells at 37 and then incubate them at room temperature, you can drive the cells in those edge wells to the inside edge of the well, which really helps solidify that this is a thermally driven effect. So just to wrap up, just remember that with constant 37 degrees C, you don't waste any of the plate or any of your money that you're spending on these plastics. You can keep the cells also at optimum full time. You know, cells get stressed when they're at the wrong conditions, and you can get perfectly reproducible conditions every assay, experiment after experiment, and be from lab to lab and location to location. So this is really going to help reproducibility for your cell-based assays and your new approach methodologies. I will stop here and thank you for your time and attention, and say that if you'd like to ask a question, if you'd like to have us talk to other people in your lab or in your company, please reach out. My email is right there. Join our groups on LinkedIn. I own In Vitro Reproducibility and Physiologic Cell Manufacturing, and we'd love to have your voice in on those groups. Thank you.